Welcome. This video is going to be on using plant sap analysis to develop an optimal nutrient profile for Lowbush blueberries, a SARE grant research project by Meredith Wecker.
Hi, my name is Meredith Wecker, and I'm a co-owner and manager of the Benson Place Farm in Heath, Mass. We've been managing more than 50 acres of certified organic Lowbush blueberries. The blueberries have been managed commercially for about 70 years here on the farm, and for the past 24 years, the fields have been primarily in organic production. I have a background in nutrient-dense farming, both from my undergraduate studies as well as from my previous experience growing vegetables. When we bought the Benson Place in 2011, I was excited to apply nutrient-dense principles to Lowbush blueberries. So, what is nutrient-dense farming? Literally, it is defined as farming in a way that results in a crop that is more dense with nutrients. I also define it as balancing plant nutrition and using management practices to maximize the plant's genetic potential to resist disease and insect pressure and produce high yields of high quality crops. So, what do optimal Lowbush blueberries look like? Well, first and foremost, we're looking for high quality fruit. That's what our customers are looking for. And for us, that means four things. First, we're looking for fruit that ripens evenly. And this is especially important for Lowbush blueberries because we only come through with the rakes once and we harvest all the fruit, ripe and unripe. The unripe berries get sorted out, and so more even ripening is going to result directly in higher yield. We're also looking for berries that have a firm skin. This has been linked to calcium levels. So calcium has been shown to result in a denser skin that's less susceptible to insect damage, especially from ovipositors like the blueberry maggot fly and spotted winged Drosophila. So berries with firm skin are also less likely to be damaged while sorting, which prolongs shelf life. And I'll also mention copper here. Copper governs elasticity of the skin. So when you get those thunderstorms at the end of the harvest and all your berries crack, having adequate copper can help prevent that. We're also looking for berries with high brix and great flavor. So having an ample supply of the nutrients necessary for the photosynthetic process helps to optimize sugar content. We also want nutrients available in ample amounts to build desirable flavor compounds. And finally, nutrient-dense Lowbush blueberries should have a higher percentage of large berries. Large berries don't get sorted out in the sorting process, and they can be sold as grade A fruit. So in unirrigated fields, I think rainfall is the biggest factor impacting berry size, but I don't want to discount nutrition. It can still play a role. For example, potassium is responsible for moving moisture into the fruit during the fruit fill stage, swelling it, and so having adequate potassium levels is important for maximizing size. And with potassium, there's the hypothesis, it appears that manganese levels, having adequate manganese can act as a type of thermostat or a regulator for potassium in the plant sap. So here on our farm, as far as Lowbush blueberries are concerned, the main disease pressure is coming from mummyberry. And the main insect pressures are coming primarily from the blueberry sawfly larvae that defoliates the leaves during the bloom time. We also have minor pressures from spotted winged Drosophila and from the blueberry maggot fly that lay their eggs in the ripe fruit. So we're looking to boost plant health in the blueberries in order to minimize these pressures also. And there's several, so as far as yield goes, we're looking for high yields, of course. There's several factors influencing differences in yield between blueberry plants in a field, including pollination timing, the timing of ripening as it relates to when we come through that particular field, and the genetic ability to uptake nutrients. So this last factor is what I was especially interested in for this SARE research project. Okay, so on our farm, as part of our nutrient-dense approach, we were amending the soil with granular amendments. There is a relative lack of data on optimum micronutrient levels in the soil for Lowbush blueberries, but we were using general guidelines provided in Gary Zimmerman's The Biological Farmer, as well as annual soil testing and recommendations through Logan Labs. In 2014, we began using foliar spray products from Advancing Eco Ag. The idea behind the foliar sprays is that you can apply an ongoing supply of nutrients throughout the season in small quantities, and that the nutrients are absorbed through the leaves and go directly into the sap. This ongoing supply of nutrients also stimulates a boost of photosynthesis in the plant, which has positive effects. On our farm, we try to get all the fields sprayed every 2 weeks, which is a huge job because we're using backpack sprayers. And initially, we were using formulas for the different stages of plant growth, pollination, and fruit fill put out by AEA. And we felt that we were getting good results. Most of all, we noticed firmer skins and more even ripening, which resulted in faster times to sort boxes of blueberries on the sorting machines, which was a savings in labor costs. And also, we noticed a vast reduction in blueberry sawfly larvae. In about 2018, we started using sap analysis through Nova Crop Control in the Netherlands. Sap analysis measures the nutrient levels as well as total sugars, pH, and electrical conductivity of the pressed leaves. We collect samples of both new and old leaves, send them to the lab, and they analyze the sap. It provides real-time data about what is available to the plant that week, and for certain nutrients, you can look at the differences between the new and the old leaves, and you can assess which nutrients the plant might be using more readily and which nutrients the plant might be saying it has too much of. When you get your results back, they're compared to an ideal result specific to your crop. So it allows for a regular adjustment of your nutrition program throughout the season to respond to the actual state of health of the plant. And this can be really helpful too if you're having a specific disease or insect problem. You can collect sap, send it in, and just immediately adjust your nutrition program to respond to pressures that you're experiencing in real time. So for us, we see it as a perfect complement to foliar sprays because both are kind of this quick fine tuning, they allow for adjustment throughout the growing season. So I think of like the soil is kind of like the baseline, and then the foliar sprays with the analytical tool of sap analysis is kind of just the way to kind of constantly tweak the crop nutrition through the season. In addition, if you're trialing a new product, you can also determine how the foliar sprays you're using are getting absorbed, and how the plant is responding, to the new product that you're using, by collecting the leaves and looking at the sap, about a week after applying the product. So we were really excited to be using these tools, but we soon realized that there wasn't any data available for Lowbush blueberries. And our results were being compared to ideal levels in sap for high bush blueberries. For certain nutrients like manganese, actually for a lot of nutrients, we were way off the chart. And this was starting to get us into a bit of a problem with our organic inspectors, who thought that we were spraying micronutrients unnecessarily, and micronutrients for organic certification are a restricted input. So that was kind of how I became curious to gain more insight into an ideal sap analysis for lowbush blueberry plants. And that inspired me to apply for a Northeast SARE grant in 2020. And we were fortunate enough to be able to carry out the research in 2021 and 2022. So the goals for my SARE grant project were to use SAP analysis to identify key macronutrient and micronutrient variations in lowbush blueberry plants that display both very high and very low levels of production, disease resistance, and fruit quality, to determine the extent to which production, disease resistance, and fruit quality in lowbush blueberries can be increased by foliar applying key macronutrient and micronutrients to different larger field plots containing genetically diverse plants displaying a mix of high, low, and average production, disease resistance, and fruit quality, and to take the first steps in creating an ideal sap analysis profile for lowbush blueberries. The hope would be for a useful working profile that could inspire and be refined by further research. So before we get into our actual project, I'm going to just talk a little bit about reading a SAP analysis report. You can see here the different nutrient levels, minerals on the left, and the current level of the new leaves is on the top and of the old leaves on the bottom. And then we have the Nova Crop Control's range for what is optimal, with a little graph for each nutrient. So the first three things listed, total sugars, pH, and electrical conductivity. I'm not going to get into talking about those too much, but they do have an influence on especially insect and disease susceptibility. And if you're interested in this, I would recommend going to the Kind Harvest website, and there's a great video course on that website, really delving into sugar, pH, and EC, and just looking at how that influences plant health. So for this project, we're looking at on the SAP analysis, nutrients that are highly mobile, and those are minerals like nitrogen, phosphorus, potassium, magnesium, And for those minerals, we want to see slightly more in slightly higher levels in the old leaves when compared with the new leaves. So that is going to tell us that the plant has enough, and that it's putting what it wants to in the new leaves. The basic idea behind it is that the plant is trying to put optimal amounts of mobile nutrients into the new leaves at all times. And so if there's a deficiency, that deficiency is going to show up in the old leaves first. And if there's an excess, it's going to put, you're going to see much higher levels in the old leaves. So you want to see ideally slightly higher levels in the old leaves, which says like the plant has enough to put what it wants in the new leaves, but it doesn't have so much that it's trying to kind of dump it all into the old leaves. So next we can look at calcium, manganese, and boron, which are highly immobile and tend to sort of settle out over time into the old leaves and then not necessarily be available. The plant can't necessarily put those nutrients back into the new leaves quickly. And so when we're looking at sap analysis for those nutrients, we're really looking at wanting to make sure that the new leaves stay consistently in the optimal range, because the plant's not going to be able to easily draw from the old leaves. So then we're looking also at nutrients that are going to be somewhat less immobile than calcium, manganese, and boron, but definitely not super mobile. So that would include copper, zinc, iron, cobalt, molybdenum. And for those, we don't want to see a huge difference between the new and the old leaves. Those levels are also going to kind of settle out into the old leaves over time. But we don't want to see like a huge disparity. And then we also want them to be in the optimal range for that crop, for the crop that we're looking at. And I should also add that certain crops, so the levels of certain nutrients are going to change through the season. So the range is actually going to be pretty wide. So for nutrients like potassium, in Lowbush blueberries, we're not seeing a big change throughout the season, but calcium is going to really accumulate throughout the course of the year. And manganese, boron, sodium levels really tend to spike in the fall. So it's not really a static thing. It is really going to change throughout the growing season. So our project was divided into 2 phases. The first phase, we were looking at the best and the worst plants in our field and trying to come up with a nutrient profile that we were shooting for as far as the SAP analysis. So we collected SAP analysis from the 10 best and the 10 worst plants in the fields for each of 4 factors. First, we scouted the field, and then we took quantitative measurements. So we were looking at high and low rates of mummyberry infection. We were looking at high and low blossom density areas and high and low yield areas, as well as high and low berry quality. And by areas, really what I mean is individual clones, like the blueberries, they, sort of have the same genetic diversity as oak trees in the forest, each clone or genet is going to be genetically different from all the ones around it. So we were looking at those plants and collecting leaves from the best and the worst in the field. So for high and low berry quality plants, we were looking at, we developed a point system scale that looked at even ripening, berry size, bricks, and skin firmness. For even ripening, we were just counting ripe and unripe berries on the stem. For berry size, we were taking the weight of 100 berries. We had a refractometer for measuring bricks and a handheld penetrometer for measuring skin firmness. And then we looked at the nutrient differences between the best and the worst plants to develop a rough picture of what sap levels we were shooting for at different times of year. So the first thing we looked at was the plants in the field with the highest blossom density and with the lowest blossom density. And interestingly, we didn't find a lot of variation in nutrient levels between high and low blossom density areas. I would be curious to look at this further in the future, but for this project, we didn't find differences for blossom density. So when it was mummyberry season, during the bloom, it was an exceptionally dry bloom time, and we barely had any mummyberry in our entire fields, which was wonderful for us, but not so great for the Sarah Grant. And we combed the fields looking for plants with mummyberry, and we did find some that were in especially wet areas. And we took samples there. So I don't have a lot of confidence in these results. I know that the folks at Advancing Eco Ag have found that mummyberry in high bush blueberries seems to diminish when calcium and manganese levels are adequate. We did not find this, we did not get this result. The calcium and manganese levels were very similar for plants that were susceptible and plants that weren't. And in fact, the manganese levels were higher for the plants that had higher levels of mummyberry. So we didn't find that result. I did notice one thing, which was that the sodium levels for the plants that were resistant were much lower compared to the plants that were susceptible. However, given that most all the plants in our field didn't have mummyberry, it's hard to know that we were actually choosing plants that had high levels of disease resistance, because we had no way of determining that. So the sap collection for high versus low quality plants were collected during the harvest, and we marked areas with high fruit quality, and then used our analytical tools to measure the bricks and weigh the berries, skin firmness, and ripening. And then of all the plants that we had field scouted, the 10 best and 10 worst, actually 2 samples of the 10 best and 10 worst were collected. And we did find some differences here. The potassium levels, calcium levels, sulfur, and manganese were the nutrients that showed the most difference between high quality and low quality plants, and to a lesser extent, the nitrogen levels as well. The high yielding plants and the low yielding plants also displayed differences in nutrient levels that were very much of interest to us. Potassium, again, and calcium, both significantly higher in the high yielding plants, especially in the new leaves, and manganese also much higher in the high yielding plants. Also, we see sulfur again higher, and nitrogen, and also phosphorus, as well as boron and silicon, are all higher in the highest yielding plants. And sugar levels aren't on this graph, but we also saw a much higher sugar levels in the high yielding versus the low yielding plants. So here we have a series of graphs that isolate the different nutrients across the different assessments. So I'm mainly going to be looking at the yield and the berry quality, the high yield and low yield, and high quality and low quality graphs, because the mummy berry, like I said, I don't have a lot of confidence in my data, and we didn't see big differences in the numbers for blossom density. So first is potassium, and again, just looking at the differences between the yield and the quality in terms of levels of potassium. Here we have calcium levels across the assessments, and again, really striking difference between the highest yielding plants and the lowest yielding plants, especially in the new leaves. Nitrogen levels, we're looking at, again, a difference between especially the new leaves in the high and low yielding. We also see a difference here in the blossom density for nitrogen, and the high and low quality as well. Sulfur, we see high and low yield differences as well as high and low quality differences. Phosphorus, especially in the new leaves for the high and low yielding, and also for the high and low quality, we see differences there. Silica levels, again, same thing, high and low yield, high quality and low quality. And silica levels, I've been told, are related to microbiology levels in the soil. Manganese, this was one of the most interesting ones for me because we're looking at quite high levels of manganese in the sap, you know, 700, 800 parts per million in the highest yielding plants in the field, which is orders of magnitude more than the optimal levels given for high bush blueberries. And this was one of the main areas of confusion for us prior to this SARE grant. So it was really interesting to see that the plants that were high yielding were absorbing more manganese from the soil than the plants that were low yielding, and similar with high quality and low quality. Interestingly, it also shows high plants that were susceptible to mummyberry were also higher in manganese, but again, I'm not having a lot of confidence in that data. Here we have zinc levels, slightly higher in the high yielding plants, about twice as much zinc in the high yielding plants as the low yielding plants. So after we looked at the best and the worst plants in our fields, we were able to come up with, a rough goal of what we were shooting for different nutrients in the sap, and we were ready to start on phase 2 of the project. So the central questions for phase 2 were, can we come close to replicating the sap analysis from the best plants in our fields by using foliar sprays on genetically diverse plots of plants, basically quarter-acre plots? And do these plants then exhibit the same high disease resistance, berry quality, and yields as the best plants? So we had 16 quarter-acre plots, 4 plots of each type, 3 treatments plus a control in 4 different fields. We selected the fields to be areas with relatively consistent topographical features. So consistent slope, or level ground, and areas with like similar amounts of weed competition. And so the plots were quarter acre. We had a control area. We had a base spray. We had another treatment that was the base spray plus extra manganese. And we had a third treatment that was the base spray plus extra zinc. The base spray consisted of nutrients from Advancing Eco Ag, trace amounts of nitrogen, phosphorus, potassium, calcium, and magnesium, as well as micronutrients, manganese, copper, zinc, iron, boron, and trace amounts of cobalt and molybdenum, plus C-stim, which is a product, a seaweed product, and C-crop, which is a product made from seawater that contains, trace amounts of micronutrients, trace nutrients and micronutrients from the ocean. So we sprayed plots once a week, and we collected leaves for sap analysis about every 2 weeks. We began treatments in 2021, about a month after plant emergence. So we were starting with pruned fields, plants that had been burned that year, and we concluded spraying the foliar sprays about 3 weeks before the 2022 harvest. So that would have been early July of 2022. And obviously we didn't, we stopped spraying in about early October of 2021 and then resumed spraying in May of 2022. So plots were measured for mummyberry infection rates, bloom density, berry quality, and yield. And we use the same measurement methods, the point system scale, as we did in phase 1 for berry quality. So we're going to just go through the different graphs looking at the SAP analysis over time. And for each nutrient, we have in blue the control young leaves, and in dark blue the control old leaves. The base treatment, which is a light red for the young and a dark red for the old. The base plus extra manganese in green, light and dark, and in purple for the base plus zinc. So for the first graph showing potassium levels, we didn't see a big difference across treatments. The lines are pretty similar, pretty close together, and not too much to say there, which is of its own interest, because here what we're looking at is what nutrients seem to be getting well absorbed through the leaves, and what nutrients the plants might be pretty adept at, getting themselves from the soil. So for calcium, we're looking at a general trend of all of the samples rising through the 2021 season, peaking in early October, and then again starting low in May of 2022 and rising up through the harvest. So the samples with the highest levels of calcium look like the young leaves for the manganese trial, the base plus the manganese, and the young leaves for the base plus the zinc. Those were high towards the end of 2021 and also towards the end, towards the harvest of 2022. But again, the lines are fairly close together, following a fairly consistent pattern. And we see the control in 2022 is somewhere in the middle of calcium levels, and in 2021 we see the control lower down. The young leaves are kind of at the bottom of the graph. So that's really interesting for me because calcium is clearly a nutrient that we're trying to boost. Our best plants, had, over 2,000 parts per million. We even had a high yielding set of the 10 highest yielding plants with over 3,000 parts per million calcium. So boosting calcium is going to be really important. And here we're showing that at least what we were applying wasn't affecting calcium levels significantly. Magnesium levels, again, we're seeing higher levels in the base plus manganese trial, higher levels in general in the young leaves, but not significant differences between the different treatments and control. Here we have sodium. The lines are fairly close together, although we do see the control at the bottom, probably due to the C crop product that we're adding and in the treatments. You can notice the spike that we have in the fall. There's a sodium spike, and the control is kind of, following that same pattern of accumulating sodium in the leaves right before leaf fall. Nitrogen, we're not seeing a lot of differences in nitrogen, or rather some not clear patterns in nitrogen absorption through the leaves. So for chloride, I was really interested because I had learned from my consultant that chloride levels should stay lower than nitrogen levels. And we actually had at different times during the project, chloride was definitely higher than nitrogen. The folks at AEA have noticed a correlation between chloride levels and nitrogen levels, where when chloride levels are higher than nitrogen, they see an increase in insect pressure, especially sucking insects. And so we see a general pattern with chloride getting high, even in the control getting higher than nitrogen at times. But the control is the lowest in chloride. So I think that that's something to be always watching and mindful of when we're using foliar sprays, is, we don't want chloride levels to get too high. So for sulfur, we see higher levels in the base plus manganese in 2021, and base plus zinc. And then again, in 2022, we're seeing higher levels in the zinc trial, base plus zinc, as well as the base plus manganese. And I should say here that we weren't adding any sulfur. This would have been just differences in the plant sap without any additional sulfur being added. Phosphorus, following a general pattern, we're seeing higher phosphorus in general in the new leaves. Silica levels followed a similar pattern across the treatments and control throughout the trial, with the base trial as well as the manganese being slightly higher. Iron levels were interesting to me because we see the control with lower levels of iron pretty consistently throughout the sap collection. And the zinc being quite high towards the end of the project. So manganese was interesting to me because the highest manganese levels were actually seen in the treatment with the base spray plus the zinc. The trial where we added extra manganese, we're actually seeing similar levels to the control. So yeah, this was extremely interesting for me. It was interesting for me to see that the control plants had quite high levels of manganese on their own, and that it seems to be that the addition of a little bit of zinc, extra zinc, was, resulted in the highest levels of manganese, whereas the addition of extra manganese didn't. So the zinc levels were consistently highest in the zinc treatment, so the base fertilizer plus zinc. Control levels pretty consistently at the bottom. We see the spring of 2022, the levels are all starting from about the same place again, despite the fact that the zinc was really high in the fall. But then the zinc peaks again at very high levels, over 30 parts per million, as we approach the blueberry season. And then we see it fall off, for the July 12th SAP analysis. We see it go back down, indicating that the plant is actually using that zinc that we're giving it. The levels spike around June 27th, and then that zinc is actually seems to be getting used by the plant, because we see the levels go back down in mid-July. With boron also, we see the treatments higher than the control pretty consistently throughout the project, indicating that boron is getting absorbed through the leaves in this form. With copper also, we see the same pattern, the treatments following a general pattern with the control consistently lower. So with molybdenum, we see again control lower, and we see higher levels in the zinc, the base plus the zinc, as well as the base plus the manganese. Aluminum levels, we generally want to see low, and there doesn't seem to be significant differences between the trials and the control, the treatments and the control for aluminum. So for cobalt, we again see the control generally lower than the other lines, with the base plus the manganese higher in 2021, and the base plus the zinc with the highest levels in 2022. So we were able to perform statistical analysis on the data we collected around berry quality from each of the 3 treatment plots plus the control. And we used analysis of variance to determine the differences in berry quality. We had 48 samples for each treatment group and the control. So for berry weights, we had the highest average weight for the control and the base, and lower weights for the manganese and zinc, with p-values, significant p-values between the manganese and base, zinc and base. Brix, the highest Brix readings were for the zinc trial, which was the base spray plus the zinc. Next was the base, followed by the control, and last the manganese trial with the base spray plus extra manganese. We had significant p-values between the zinc and control, and the manganese and the base, and the zinc and the manganese. For ripeness, we didn't see significant differences between the treatments and the control. And for firmness, the highest readings were in the base spray plus extra manganese, followed by the control, and then the base, and finally the zinc, with significant p-values between the manganese and base, and zinc and manganese. We weren't able to perform statistical analysis for our yield data because the 4th field, which is called the owl, was completely eaten by bears and became also overgrown a little bit with weeds. And we weren't able to harvest there at all. So we did not have yield data from the owl, and therefore we weren't able to analyze the yield data. So key research findings for this project. First, there are genetic variations in nutrient uptake ability and capacity for nutrient uptake between the highest and lowest yielding plants in the field and the best and worst quality berries in the field, as seen in the data in phase 1. Secondly, most of the macronutrient sprays, nitrogen, magnesium, potassium, phosphorus, and calcium, don't seem to be well absorbed through the Lowbush blueberry leaf. These were the sprays we were using, and it could be that the plants just weren't responding to it, but given that specifically things like calcium, the plant was indicating it could use more calcium, but yet we weren't able to get the calcium levels up, indicates that it probably was an absorption issue. The micronutrients, on the other hand, do seem to be well absorbed through the leaves. Zinc, copper, cobalt, boron, iron, and molybdenum all were much higher in the treatments than in the control. And finally, Lowbush blueberries seem to be relatively efficient at manganese uptake. And that was a really interesting to see that even in the control, the plants were able to get quite a bit of manganese into their sap without any additional input. The addition of extra zinc to the base formula, so going from 1 pint of solution to 1 quart of zinc solution, which translates to actually just kind of fractions of an ounce of zinc per acre increase, seems to relate to nutrient releases of calcium, iron, manganese, zinc, sulfur, and cobalt. And this would be, just kind of a preliminary correlation and something to look further into. Optimum sap analysis in quarter acre plots was not easily replicated. So we had our levels that we were targeting, and we weren't able specifically to get the calcium levels, potassium and nitrogen up to the levels that we were seeing in the very best plants in the field. And finally, I learned that when we're using foliar sprays, we do really need to watch chloride and sodium levels, as well as electrical conductivity, and just be mindful that those levels aren't getting too high in the plant. So Nova Crop Control says that they typically will collect data on 1,000 samples before they're ready to kind of issue optimum levels for a crop. So this isn't by any means definitive, but this is just our best attempt at coming up with kind of a starting place to be further refined, and but something that's actually for low bush blueberries, instead of just comparing the levels to high bush blueberries. These are levels that we're looking at for during the harvest time. So the calcium levels being at that high, you wouldn't get those levels in the spring, but this is more like late July, August levels. And so I'll just go through them. Potassium, 1,800 to 2,400 parts per million. Calcium, 2,500 to 3,500. Magnesium, 1,100 to 1,400. Sodium, 7 to 15. Total nitrogen, 350 to 450. Chloride, keeping it lower than total nitrogen. Sulfur, 450 to 600. Phosphorus, 350 to, I wasn't able to come up with an upper range for phosphorus, and same for silica. I think having silica levels at least 30, but like in our best plants, we were seeing levels of 65. So not sure what the upper range is there. Iron, 3 to 7 parts per million. Manganese, 500 to 800 around the time of the harvest. Again, in the spring, the levels are going to be a lot lower. Zinc, 12 to 15 parts per million. Boron, 12 to 20. Copper, 0.5 to 1. And then I wasn't able to make any guesses on molybdenum, aluminum, or cobalt based on our data collection. So we did have some issues with the research. I learned a lot about designing research projects through this SARE grant, and my first time doing on-farm research. And so here's some of the issues we had. So first of all, sap collection requires a large area in order to frequently collect leaves. So normally we're collecting over the span of like an entire field, and we're collecting, maybe 4 times in a season. So over collecting the leaves is not an issue. But for the SARE grant, we were worried if we're collecting leaves every 2 weeks on plots, we don't want to be taking all the leaves off the plants. And so we ended up deciding to go with 1/4 acre plots, but these were gave less accurate results for yield data because the plots had, pretty different rates of plant coverage as well as weed pressure. We tried to keep them consistently weeded, but being an organic farm, it was, we did notice that there was differences in weed pressure. There was also differences in the amount of bedrock between different plots that I think made the yield data not very accurate. And smaller plots would have been a lot easier. Possibly we could have done less frequent sap analysis on smaller plots. So there was this kind of tension between wanting the accuracy that would come with a smaller plot, but needing a large plot in order for the frequent sap analysis. So that was tricky. On a smaller plot, also, we took 48 samples per section for the different aspects of berry quality. And on a smaller plot, this would have been, more representative of the entire plot. So another issue is that sap analysis is time consuming and expensive. Ideally, we would have performed sap analysis every 2 weeks on each of the 16 plots, but instead we needed to pool the different treatments each time. So we collected leaves from, all of the different controls and then all of the different base sprays like that into one sample in order to get enough leaves. This wouldn't be an issue with other crops, but for Lowbush blueberries, the leaves are so small, and to get enough material for the sap analysis, we needed to pool the samples. And also, we would have done more samples from the best and worst plants, but because of the time involved and the expense of the sample, we weren't able to do this. The owl field, as I mentioned, got overgrown with weeds to the point where, I think a lot of the sprays were ending up on the weeds. And then the berries the field was completely eaten by the bears, so we weren't able to collect we weren't able to perform statistical analysis on the sap data or the yield data. It would have been interesting to spray right up until the harvest, but because we didn't want to step on the right berries, we weren't able to. Maybe this is a problem, but it's also kind of representative of what we normally do anyways. And then finally, because we've been using these sprays for several years on our farm, we didn't have any true controls. We didn't spray the control plots that year, but the plots had been sprayed in previous years. And so that kind of also just calls into question, how much is sort of accumulative especially with the macronutrients, and what would it be like if we were doing this experiment on a plot with that had never been sprayed, or plots that had never been sprayed. This SARE grant project has given me a lot of ideas and inspiration for further research. Of most interest is performing more SAP analysis on the best and worst plants from many fields all over in Maine, southern Canada, fertilized and unfertilized. I think being able to collect a pool of SAP data would really further our understanding of optimal levels. The goal would be to create an ideal SAP analysis picture for different times of year. So, what would it ideally look like in the spring, pollination time, as well as early fruit fill, late fruit fill, post-harvest, and leading up into the fall. I'm excited to trial new macronutrient products and to see if we can get different products that will absorb well through the leaves, see if we can get a response, especially with calcium products. There's a company called Ferticell that makes different macronutrient sprays, and interested in looking into those. They also make a product called Nutra Plus that's an adjuvant and is supposed to help other sprays become better absorbed. It's an amino acid product, and I'm interested in trying that with the AEA macronutrient sprays this year and seeing if I can get a response on the SAP analysis. I'd really like to look at SAP analysis for mummyberry susceptibility over several years and on various farms. I'm really curious about, what they found with highbush blueberries with getting calcium and manganese levels optimal, and seeing if that, is true for lowbush blueberries or if there's a different set of factors. That part of our research, wasn't fruitful, as it were, in terms of really looking at what the correlations were. So I'm still curious about that. And then again, like I mentioned before, really interested in what it would be like to do this experiment again on previously unfertilized fields. I would like to express my gratitude and appreciation to the following people and organizations for their help with this project. First, Northeast SARE for providing funding and support, and Candace Huber as my SARE coordinator. Nathan Harmon, my technical advisor. Nick Casal at AEA for support with my numerous questions. Maisha Stevens for heading up data collection during the 2022 blueberry harvest, which is a busy time of year. Satya Benson for performing statistical analysis. Julia Zeku for making graphs and putting in long hours to make this slideshow presentation. And Andrew Karowski, my partner and co-worker, co-manager, for his many hours of support with computer work, patience, and understanding. Thank you to everyone.